Mitotic cell division alternates with a much longer stage called the interphase, which often accounts for about 90% of the cycle. During interphase, a cell that is about to divide grows and copies its chromosomes in preparation for cell division. Interphase can be divided into subphases, the G1 phase, the S phase, and the G2 phase. In G1, a cell grows. In S, it continues to grow as it copies its chromosomes, and in G2, it grows more as it completes preparations for cell division. Mitosis is broken down into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase.
Mitosis is a division of somatic cells and allows for growth, regeneration, and reproduction.
In G2, we have 2 duplicated centrosomes, which organize the mitotic spindles. Also present are the duplicated chromosomes contained in the still intact nuclear envelope. We cannot distinguish them because they have not yet condensed. Now begins prophase. The chromosomes condense and appear as sister chromatids joined at the centromere. Centrosomes begin to move to the poles as microtubules begin to form. The nuclear envelope fragments, allowing microtubules to enter the former nuclear space and attach to the centromeres at kinetochores. Kinetochores are protein complexes located on the centromeres, which allow for the attachment of microtubules. Kinetochore microtubules begin to tug and pull on the chromosomes, pulling them into correct alignment.
During metaphase, the centrosomes are now on opposite sides of the cell. The chromosomes align on the metaphase plate, a line equidistant from both centrosomes. The connectochores of each sister chromatid are attached to the connectochore microtubules from each side of the pole. Next is anaphase, which is the shortest stage of mitosis. The cohesion proteins are separated and each chromatid becomes a full chromosome. The 2 daughter chromosomes begin moving towards opposite ends of the cell and the microtubules shorten. Non-connected core microtubules elongate and lengthen the cell.
Chromosomes become less condensed, and the nucleus begins to reform. The microtubules have become completely depolymerized. A cleavage furrow forms from the microfilaments, splitting the 2 cells. The result is 2 genetically identical daughter cells. This indicates the end of cytokinesis and mitosis. Now on to meiosis. Meiosis has 2 major components, meiosis I and meiosis II. Essentially, meiosis I is a separation of duplicate homologous chromosomes, and meiosis II is a separation of sister chromatids. Meiosis is essential for genetic variation among sex cells. It results in 4 haploid cells.
During prophase, the chromosomes begin to condense and homologs pair up and become physically connected to each other. This is called synapsis. Crossing over then occurs in which corresponding segments of DNA molecules switch between the 2 homologs. Each homologous pair of chromosomes has 1 or more X-shaped regions called chiasmata. This is a site where crossover has occurred. Centrosome movement, spindle formation, and nuclear envelope breakdown also occur. Later in prophase, microtubules then attach to the 2 connectochores at the centromeres of the 2 homologs. In metaphase, the pairs of homologous chromosomes are arranged on the metaphase plate with 1 chromosome attached to a connectochore microtubule facing each pole.
During anaphase, the homologs separate due to decomposition of proteins responsible for cohesion. Homologs are pulled to opposite poles by microtubules. During telophase and cytokinesis, we see the creation of 2 diploid cells containing duplicated chromosomes with regions on sister chromatids containing non-sister chromatid DNA. A cleavage furrow forms, splitting the 2 cells into 2 genetically different diploid cells. In some species, the nuclear envelopes form and chromosomes decondense.
In prophase, a spindle apparatus forms. In late prophase 2, chromosomes are still composed of 2 chromatids associated at the centromere. Chromosomes are positioned at the metaphase plate as in mitosis. Because of crossing over in meiosis I, the 2 sister chromatids of each chromosome are not genetically identical. The connectochores of sister chromatids are attached to the microtubules extending from opposite poles.
Anaphase II is a separation of sister chromatids. Decomposition of proteins at the centromere allows separated chromatids to move towards centrioles at the poles as individual chromosomes. During telophase II and cytokinesis, nuclei form, the chromosomes begin decondensing, and cytokinesis occurs. The meiotic division of 1 parent cell produces 4 daughter cells, each with haploid set. The 4 daughter cells are genetically distinct from one another and from the parent cell. Each of the 4 cells contains only 1/2 of the chromosomes represented in the original parent cell. There are several differences between meiosis and mitosis. Mitosis is a single division of a somatic cell to produce 2 genetically identical daughter cells, with a chromosome count of 2n. Meiosis is a process which involves 2 divisions, the first to separate homologs and the second to separate sister chromatids. Meiosis occurs in sex cells and some bacteria cells. Meiosis produces 4 genetically different cells through independent assortment. Each of the 4 daughter cells are haploid with a chromosome count of n. An example of mitotic cells in the human body are sex cells, or gametes.